Okay, so my name is Kristen Buck. I'm a chemical oceanographer here. I specialize in trace metal biogeochemistry, and our lab is particularly interested in the role of organic ligands. So I'm going to give you kind of an overview of the work that we do in my lab and the background behind it. Those of you who have seen me give this talk before, it's not too different. So I hope you're all familiar with the periodic table. Generally, this is kind of the standard look where it's color-coded by the type of elements. This version of the periodic table is colored according to the abundance of the elements in seawater. I'll find my little thing here. Yep, all right. And so I didn't make this particular map, but in this case they did highest concentrations in the blues and purples, the cool colors, lowest concentrations in the warm colors, the red and orange, and you can see sodium, chloride, the major components of seawater are very abundant, as you would expect. The transition metals here in the middle and the trace metals are the ones that my lab's particularly interested in, are here in green. And each one of these color blocks denotes a 1,000-fold decrease in concentration. And so these elements are roughly 1 billion times less abundant than the major components of seawater, and their concentrations are typically on the order of parts per trillion. So their concentrations are very low. In molar units, these elements are typically on the order of nanomolar, 10 to the minus 9 molar, to picomolar, 10 to the minus 12 molar. So this presents analytical challenges, you can imagine. We also go out and work on iron ships covered in copper-based anti-fouling paints, which presents sampling problems as well. And these elements are important because their bioavailability, the amount of the element that is available to the biological organisms in the ocean, actually governs how productive the oceans are. And so they have the most influence on the smallest organisms, which are the phytoplankton. These are the plant-like base of the food web. And you look at, this is a table derived from Morell and Price, and it shows the biogeochemical processes and then the trace elements involved in them. You're talking about carbon fixation, which is photosynthesis, uptake of different nutrients, whether that's carbon, nitrogen, cycling of nitrogen, phosphorus, and even toxicity. And I put up some pictures of the pretty phytoplankton so that you could remember that these are very small organisms. These are diatoms over here on the left. They tend to be of the largest phytoplankton groups, phytoplankton species in terms of their size. Coccolithophore is here. This is trichodesmium, a nitrogen fixer that lives in the surface ocean. And I underlined in color iron and copper. So iron in the red underline and copper in the blue. And while we're interested in a wide range of trace elements in seawater, iron and copper are the two that we focus most of our research on. And part of that is because you can see that they play a very wide-ranging role in governing productivity of phytoplankton. So iron in particular is a limiting micronutrient in the oceans. It's exceedingly important. This, again, is from that same Morell and Price paper. This is showing the nitrogen cycle, and then the colors are the different trace elements that are involved in the enzymes for each step of this cycle. And you can see that iron is in is in every step of the nitrogen cycle, one way or the other. Over here on the right is showing biomass. This is your phytoplankton here, looking at different nutrients, so phosphorus, inorganic and organic phosphorus, nitrogen compounds, carbon compounds. And you can see again that iron is incredibly important, particularly in nitrogen uptake. And because of this, regions of the ocean that are limited by iron tend to reflect high nitrogen concentrations in surface waters. So this is a global ocean map. So here, this is back to a color scale I'm more comfortable with. Warm colors are high concentrations, cool colors are low concentrations. And you can see that in surface waters of the global ocean, nitrate, which is an inorganic nitrogen form that is preferential for, say, diatom growth in the oceans, is high in surface waters in the Southern Ocean, the equatorial Pacific, and the subarctic Pacific. These regions are collectively known as high nutrient, lower than expected chlorophyll, or HNLC regions, and these regions are characterized by iron limitation. So almost half of the surface ocean on this planet is limited by iron, meaning phytoplankton there have enough nitrogen, phosphorus, they get plenty of carbon from the atmosphere, but they don't have enough iron to grow to their maximum productivity. So iron is exceedingly important in understanding productivity in the open ocean, and that plays into not only the nitrogen cycle, but in turn the carbon cycle. Copper, on the other hand, is what I refer to as a Goldilocks element. While most of my research is on iron, anybody who knows me knows that my dog's name is Copper, and that's probably insight into where my love falls. So hopefully you guys aren't so young that you aren't familiar with Goldilocks, because that's depressing to me. But this is the idea that, too little is not enough, too much is too much, right? You want to be just right. And this is best represented, in my opinion, in a growth response curve. So here we've got bioavailable metal concentration on the x-axis, phytoplankton growth rate on the y-axis. So how phytoplankton growth rate changes as a function of the bioavailability of that element. If you don't have enough of that element, then as you increase the bioavailability of that metal, the growth rate of the phytoplankton organism will increase. When you reach sufficiency, the just right bowl of porridge, if you will, you can change the concentration of that element and it has no effect, or the bioavailability of that element, it has no effect on phytoplankton growth. Once you reach this threshold, as you increase the bioavailability of that element, it becomes toxic and it can decrease, negatively impact your phytoplankton growth. Copper is somewhat unique in that most of the oceans fall in this green region, but where you get into inshore regions, anywhere where people are, we contribute a lot of copper into the environment through anti-fouling paints, through wastewater discharge, surface runoff. There's a lot of copper in the brake pads of your cars, things like that. And we reach this toxic threshold frequently in inshore waters. So we want to be Goldilocks. We want to be right here in the middle. As an example of the effect of copper toxicity on phytoplankton, so here we have different phytoplankton groups, diatoms, these are the biggest phytoplankton, coccolithophores, dinoflagellates, and cyanobacteria. Cyanobacteria are the really, really tiny guys. And what you can see is that this is a log scale of free copper concentration, which is a bioavailable form of copper. As you increase the free copper concentration, you reach a threshold point where the phytoplankton growth rate starts to decline. For the larger phytoplankton species, like diatoms and coccolithophores and dinoflagellates, this is a little bit later. They can tolerate a little bit more copper, and you'll learn all about that in chemical oceanography. And at relatively low concentrations of free copper, the smallest cells are susceptible to toxicity, and they will decline. So, I keep talking about the bioavailability of elements and how important this is. This is where kind of my research falls. What we do in my lab is we look at bioavailability and the role that organic ligands play in governing bioavailability. So it's not just about concentrations. The total dissolved concentration of iron and copper in seawater is a sum of the inorganic species, noted collectively here as iron prime or copper prime, and the organic species, the iron L or copper L. Now, the inorganic species, these are the most bioavailable, and specifically as the free hydrated ions. You saw that in that copper toxicity diagram. So these are really important for bioavailability, but they tend to be very, very low in seawater because more than 99% of iron and copper are present as these organic ligands, and that is not happenstance. So these organic species, they can be bioavailable. So phytoplankton are geniuses. Bacteria are even better geniuses, if you will. They will find a way to get what they need. So they can get these organically complex forms of metals if they really need them. But the free ions are very difficult to keep out of the cell. They have relatively, I don't want to say passive uptake system because I might get in trouble for that, but those free ions tend to cross into the cells fairly easily through established uptake mechanisms. Now, for iron, organic ligands actually increase the bioavailability of iron, which is counterintuitive since the inorganic form of iron is considered the most bioavailable from the biology perspective. But the reason for this is because the solubility of inorganic iron is exceedingly low in seawater. It's too low. If it was only an inorganic system, there wouldn't be enough iron to sustain phytoplankton growth in the oceans as we know it. Organic complexation, these ligands and their concentrations and their conditional stability constants, their strengths for binding iron, the stability of those complexes, drives iron cycling. I co-supervise a PhD student who just graduated from the University of Plymouth who looked at this process, and this is a little confusing, so let me walk you through it. Iron comes to the ocean predominantly in the form of particles. In the surface ocean, that's largely coming from dust. This is Africa here. You see this big Saharan dust plume. This is a common figure that we look at for dust deposition into the surface ocean. I put this other one down here so you could remember that dust influence influences much more than just the eastern Atlantic. In fact, we get Saharan dust deposition here in Florida. But as that dust comes into the surface ocean from the atmosphere, it's particulate iron, and it's got all these little inorganic iron species on it, if you will.